Okay, so now that we understand about vasoconstriction, vasodilation, and that effect that would have on total peripheral resistance, which is one of the factors that will control blood flow. When we change that equation, we saw, like in our previous video, that the peripheral resistance will affect blood pressure. All right, so let's actually go through the homeostatic process as it relates to that. And when we do that, we're going to make sure that we understand which portion of the brain is involved with controlling blood pressure. Okay, we had a chance with cardiac output to look at chemoreceptors picking up the amount of chemicals or type of chemicals you have in your blood. But now we're going to look at how blood pressure is going to be controlled in homeostasis. Okay, so let's take a picture. Let's basically look at that process. Let's remember that in homeostasis, and I'm going to do this with plenty of other systems, right? Very important to understand homeostasis and the parts of homeostasis. So we're all in homeostasis. There's always a stimulus that changes the internal environment. Okay, whatever that particular variable is, it's controlled and within normal limits. Right? So the internal environment is fine when that's the case. But if those variables are changed, that changes that control condition of that internal environment, our extracellular fluid. So therefore, because that's a change, that's going to mess things up as it relates to how we function. So we have to have detectors in our body to detect these changes, and those are called receptors. Receptors then take that signal, that information to control center. We have different types of control centers in our body. Most of the time it's the actual brain or the spinal cord, and then the actual decision is made there at the control center. And then from the control center, that information is taken to an effector. Whatever that decision was made from the control center, it's going to go to, that information is going to go to an effector that's going to cause a change, and now you're going to have a response as a result of that stimulus. All right? Typically, the response is going to oppose the stimulus. Okay, and that's the homeostatic process that we call negative feedback. All right, so let's take a peek at that, and let's give an example like we have blood pressure. If we have low blood pressure, a decreased amount of blood pressure in your entire systemic circulation, not good, right? Because you need enough pressure to get enough pressure in blood for that blood to get to tissue. All right, why does that have to get there? Because that blood has oxygen and nutrients in it, okay? So we want to make sure that there's enough blood pressure. Well, you don't have any control over it. Homeostasis takes care of everything. And so what's going to happen is that your low blood pressure is going to be that stimulus that changes your internal environment. All right, now you have what? A low blood pressure in that internal environment. Not good. Can't get oxygen and nutrients to tissue. Right? It has to be above 90 over 90 systolic, 90, okay, over 60. Okay, it has to be over that. Okay, typically blood pressure, normal blood pressure is between, okay, 90 over 60 to 120 over 80. Okay, anything below that, systolic and diastolic pressure, anything below that is considered low blood pressure. All right, that's not enough oxygen and nutrients getting to tissue, okay? So now we have our controlled condition changed, okay? The variable, that variable is not within normal limits. So who's going to detect that? Receptors. And the receptors involved, okay, are not too different than what we talked about before, and especially the location. Your carotid artery, which is a branch from the aorta, and the aorta itself has in the lining receptors of the actual vessel. And well, what are we detecting this time? We're detecting not chemicals, but pressure. And just like a barometer, that's what that detects, pressure, right? A measuring tube for pressure. So that's why we call these receptors baroreceptors. Baroreceptors. Where are they? The carotid baroreceptor and the aortic baroreceptor. They're going to detect this low blood pressure and then take that signal information to the medulla oblongata, But now, when we're dealing with changing blood pressure, we're actually going to go to vessels. There's going to be a signal taken to vessels to make a change, not to the heart. All right. So before in the medulla oblongata, we had a group of neurons that were responsible for changing things in the heart, and that was your cardioacceleratory center and your cardioinhibitory center. But not when we're dealing with pressure so much. Well, yes, it can deal with pressure, cardioacceleratory center and cardioinhibitory center. But as far as changing that pressure as it relates to vessels changing the pressure, then we're talking about a vasomotor center. And so we have all of those different groups of neurons in the medulla oblongata. We do have the cardioacceleratory center and cardioinhibitory center. But we also have a vasomotor center specifically related to changing, making changes with blood vessels. All right. So we'll see that in a second. So the vasomotor center makes a change, makes a decision, says if we have low blood pressure, you get it. At this point, homeostasis, especially the most abundant type, is negative feedback. You're actually going to have the opposite. You're going to want to do the opposite of what the stimulus is. So vasomotor center is going to send a signal to the effector. What are those effectors? Smooth muscle in arterioles, and it's going to cause them to contract. Now, if that smooth muscle contracts, just like you saw in the previous video, then that's going to cause what? Vasodilation or vasoconstriction? Well, you guessed it. It's going to cause what? Vasoconstriction. Now, if your diameter decreases, that's like Interstate 95 going to 1 or 2 lanes, what does that actually do to peripheral resistance? Yes, that actually increases peripheral resistance. And if we increase peripheral resistance, what are we doing to blood pressure? We're increasing blood pressure. Okay. So now that's perfect because our response is this actual vasoconstriction with now increasing blood pressure. And that makes sense because increasing blood pressure opposes our stimulus, which is decreased blood pressure. And now that totally brings home how this has to be that negative feedback process that we've been talking about, right? The opposite is just going to happen as it relates to increased blood pressure if that's the stimulus, and then we get the opposite response. Okay, so this is just a good look at what we were talking about as far as how arterioles can change blood pressure depending on whether they do vasoconstriction, they'll increase blood pressure, or vasodilation, which would decrease blood pressure. All right, so that's just bring home what we looked at with the last video there. Okay, very good. All right, let's just peek at our vascular scenario as it relates to arterial again, but also the capillary. And let's look at how we have these two different factors of what's actually going to be centered around your capillary bed at tissue level. Okay, at tissue, we have our capillary. And let's take a peek at what drives that oxygen and nutrients into tissue. And what that is called a hydrostatic pressure. A hydrostatic pressure, which is the fluid inside of the arterioles and the capillaries, pushing on the lining of the actual wall of these actual vessels. So that makes sense now. What is that pressure going to be doing? It's going to actually be pushing fluid out of the capillary, okay? And so where does this particular pressure come from? It comes from blood pressure, okay? Your systemic blood pressure, okay? At tissue level, we call that, when we're specifically looking at that pressure right at that capillary, we're calling it hydrostatic pressure, right? That fluid pressing on that wall of the actual blood vessel. And that's why we say hydro for water. Most of that blood, which is fluid, is water. So it's pushing on the walls of the actual vessels. Now what does that do? It pushes all of the what out? Oxygen and what? Nutrients, okay? And so that's what's great about this particular pressure, which is basically dominant on this actual end of the capillary. We call this the arterial end of the capillary, right? But it's just very close to the arteriole, okay? This is the arterial end of the capillary. And out of the two pressures that are there, there are two pressures. I'll get to this one in a second. But out of the two pressures right here on this arterial end, the one that's highest, the one that's dominating, is called the hydrostatic pressure. And that's good because now we're going to be able to get oxygen and nutrients out of the capillary and into the actual tissue where our cells can now pick up that oxygen nutrients and live. Right? But what else does your actual cells create? They create CO2, our main waste product. And now that CO2 cannot stay in cells or those cells will die. Right? So what's going to be on this end of the capillary? Well, we're going to remind ourselves that we have these actual proteins that are in the blood and are not able to get through those fenestrations as your hydrostatic pressure is trying to push it through. It's not going to. These proteins are too big for that, right? What are those proteins called? Those proteins are called albumin, okay? And yes, they are in the arterial end of the capillary, but it's not as many of them on this arterial end because you have way more water over there, okay? However, as this albumin continues to go to the venous end of the capillary, that's what we call it, the venous end, because it's close to the veins. So we call this the venous end of the capillary. It's a totally different thing there because all the water came out on the arterial end, so we don't have much water now on the venous end. So that means that changes the tonicity. You remember chapter 3 when we said that when you have a solution which is in this venous end where there's a lot of solute, way more solute than the actual solvent, then it's a hypertonic solution, right? And so that's what we have on the venous end. On the arterial end, we had a hypotonic solution. But on the what? On the venous end, we have a hypertonic solution. And then we have all this water out here in the tissue that came from the arterial end. So now where is that water going to go that's in that tissue? All that water is going to go where? Right on in. Right on into this capillary here, but on this end. Which end? The venous end. All right? Which is guided by who? Albumin. See, he's the main solute. Like we talked about in chapter 19. The most abundant solute in your actual body. Since proteins are the most abundant solutes, the number one protein in highest number is called albumin. And so he's the solute then that's responsible for pulling water into the capillary on this end of the capillary, right? And so we call that pressure, because it's related to water flowing from high concentration to low. That's what we talk about chapter 3. We call that process osmosis. So if you were just standing right there and you received all that water, you would feel a lot of pressure, right? So that's why we call this osmotic pressure. And what is osmotic pressure guided by? Albumin, okay? So on this end of the capillary, we're very, very dependent on albumin to make sure that there's that osmotic pressure there. Now different though, right? Before it was really low on this arterial end, but on this venous end, your osmotic pressure is higher and dominates your hydrostatic pressure. See, not much fluid over here, so not much fluid to press on the walls here. So which actual pressure is dominating on the venous end of the arterial, of the capillary, That's your osmotic pressure, and that's good because now your net flow of fluid will go from the tissue into the capillary. And that's great because a lot of that fluid has CO2 in it, right? And we want to take CO2 away from tissue, right? And back to the heart. And like we know with pulmonary, it goes actually to the lungs, and then we breathe it out, right? So we highly depend on albumin. We highly depend on osmotic pressure to drain our tissues. And that's what's happening on your capillary bed. You have two portions of your capillary, the arterial end and the venous end. And at our arterial end, which pressure out of the two pressures is dominating? Your hydrostatic pressure. Your hydrostatic pressure dominates on that arterial end. Now, pushing blood out to tissue, right, or fluid out to tissue, because not all components of blood come out. They can't fit through the fenestrations. So that hydrostatic pressure guides all of that fluid out and into the actual oxygen, now giving the tissue oxygen and nutrients. And then we have on the venous end, the dominant pressure being osmotic pressure. Yes, hydrostatic pressure is there, but it is not as high as your osmotic pressure. And now what happens? All of that net flow of fluid is now going to go into the actual capillary, but the venous end of the capillary, and bring with it carbon dioxide. And now you can blow it out. So that we highly depend on that, right? If you have really, really high blood pressure, then that means your hydrostatic pressure might be a little higher than usual here now, right? So that means you're dumping way more water in your tissue than you can. And what you have? The same drain rate. So that means you might have way more water in your tissue than you should. And what am I describing right there? A person who's got hypertension. A person who constantly has high blood pressure might have a lot of swelling in their tissue, which we call edema, as it relates to their high blood pressure. On the other end, you might have an alcoholic who damaged his liver. And now, where do all of your proteins come from? The liver. And our most abundant one will be in low number then, right? And if we don't have enough albumin, then you're not going to have your osmotic pressure dominate your hydrostatic on the venous end. And now you're not going to have much water doing what? Being drained from tissue. Therefore, all the water stays out here in the tissue, and that means now you have swelling again. Called what? Edema, but for a different reason. The reason is liver failure, right? Because you don't have albumin. Albumin's the cause. If you don't have albumin, then you're not going to get enough fluid drained from tissue. Right? So two different cases, and that's why clinically you just have to look at the case and see what maybe the cause is. Right? And definitely we see that's not good at all, because we definitely need to drain our tissue, don't we? Yes, very good. All right, so that's what we're looking at as it relates to the capillary, and a good look at what we consider that vascular end of this cardiovascular system.